So today we're going to record a video in preparation for the biology EOC, and it's on the molecules of life, specifically DNA and RNA. So our agenda today is to discuss what is DNA specifically. We're going to talk about how DNA pairs together, or base pairing between DNA. We're also going to compare DNA versus RNA, and we'll discuss the central dogma of biology, as well as mutations. So in preparation for the EOC, here are 4 benchmarks that are going to be covered in this video and which will also be assessed on the exam. You'll see here that I've color-coded them red, green, blue, and purple, and they're embedded at the bottom of the PowerPoint. So if you're having trouble with these specific topics, definitely go back and revisit them, as they will be assessed on the end of course exam. So starting with the structure of DNA, we know DNA stands for deoxyribonucleic acid, and it's a very large molecule. Also known as a polymer, meaning that it's made up of repeating units known as monomers. And these monomers in DNA are what are called nucleotides. That's an important word. And a nucleotide is made up of 3 things: a phosphate group, a nitrogenous base, and a 5-carbon sugar. And you'll see here in the diagram, we've got an illustration of this as a circle, pentagon, and a square. So we've got your phosphate, deoxyribose sugar, and your base. So that's the structure of a nucleotide, and DNA is made up of many of these nucleotides, and that's what's called a polymer. Now you'll see here, focusing in on just this group, the base is important because there are 4 different kinds of nitrogenous bases in DNA. And you see here, adenine, abbreviated as A, and guanine, abbreviated as C, are known as purines. And it just means they're a double ring structure. You see these 2 shapes joined together. So A and G are what are called purines. T and C are pyrimidines. Also, but T and C stand for thymine and cytosine. That only means that they're single ring structures. Now, the bases are critical in this whole understanding of DNA because they pair with one another. So base pairing is complementary in DNA, meaning that the base pair adenine pairs with thymine, or A pairs with T. Cytosine pairs with guanine, abbreviated as C, pairing with G. Now RNA is similar, but there's a slight difference here. Again, cytosine pairs with guanine, that's the same, but adenine pairs with uracil. Big difference here. And you'll see that these bases are held together by hydrogen bonds. Now, the structure of DNA is very important, and in 1953, Watson and Crick won the Nobel Prize for coming up with this structure, and the structure is similar to what we would call a ladder, kind of like a spiral staircase or twisted ladder, and Watson and Crick referred to it as a double helix. So you may hear those words on EOC day. And it's like a ladder because the sugar and phosphates make up the sides. So you'll see here, we've got phosphate group, sugar group. Phosphate, sugar, phosphate, sugar, phosphate, sugar, going down either sides of the ladder. And those nitrogenous bases are the rungs or the steps in the ladder. And you'll see here T and A, G and C, C and G, A and T are the bases that meet together like the steps in a ladder. So those bases are held together by hydrogen bonds. And even though they're weak forces, they're strong enough to hold both strands of DNA together because DNA is a double-stranded molecule. But the hydrogen bonds also allows the DNA to easily pull apart when it needs to replicate, and we're going to talk about that next. So here's a little comparison between DNA and RNA. We know that DNA stands for deoxyribonucleic acid, and that means that the sugar base is a deoxyribose sugar. If you see here on our diagram, here's deoxyribose, and it's considered a deoxyribose molecule because of that second carbon right here does not have an oxygen. So deoxy means no oxygen, and it's missing on that second carbon. It's also a double-stranded molecule. You can see that from here. We've got 2 strands held together by those hydrogen bonds, and it also contains the base thymine. Now RNA is a little bit different. It stands for ribonucleic acid, and it contains the ribose sugar, which all that means is that on this, again, the second carbon, it contains that oxygen atom, and that just makes it a ribose sugar. Now, RNA is a little bit different in that it's only single-stranded. You'll see here from our diagram, only a single-stranded molecule, and it contains the base uracil. instead of thymine. So the structure of DNA is very important because sometimes in a cell, specifically during mitosis, the cell wants to copy all of its DNA, and that process is called DNA replication. And what the cell does is it uses the sequence, which just is another word for order, of those bases on a strand of the DNA. So we said it's two-stranded, and one strand is copied. And it's because the strands are complementary, going back to our base pairing rule, A pairing with T and C pairing with G, that we get a new copy from the original. And that's what's called a template. So each side of the ladder is used as a template to make a copy of the original. And we're going to do a practice to illustrate that. So here's our example. So we've got these DNA sequence of TCG, AACT, and the complementary strand would be AGC, TTGA. And I get that by using the base pairing rules. Remember again, A pairs with T and G pairs with C. So assuming, looking at this letter T, T would pair with A, C would pair with G, and G would pair with C, and so on and so forth. Now I'd like you to try this. What would be the complementary sequence of DNA to the molecule below? ATT, CGT, AGG, ACT. So by using our base pairing rules again, we know A pairs with T, T pairs with A again, G, C, A, T, C, C, T, G, A. So that would be the complementary sequence. Again, this is a benchmark, so I would definitely review this concept. It most likely will be assessed on the EOC. Now, the central dogma of biology. So we said sometimes during the cell they want to copy all of its DNA, but at other times it just wants to copy small portions because DNA codes for proteins. And this is what's called the central dogma of biology. Dogma just being a belief system. So scientists believe that the DNA, which codes for RNA, and is made into a protein. And it's done by these two steps. And they're very important words. I would starring them because they're critical words, and those words are transcription and translation. So this process of the DNA being transcribed into RNA is what's called transcription. And eukaryotic cells, it takes place in the nucleus. You can see here we've got a double-stranded molecule. It's copied into a single mRNA copy. Again, that's called transcription. Now, once that mRNA leaves the nucleus, it will be translated into a protein. So this process of the mRNA being read into a protein sequence is what's called translation. And we're going to illustrate this a little bit further in just a moment. So once the cell has that mRNA copy, it's been transcribed from the DNA, we know that there is a genetic code embedded in there. And that code just refers to a series of 3 base pairs known as a codon. So you can see here's our mRNA molecule, and it's sequenced in bases of 3, which are referred to as codons. So codon 1 is AUG, codon 2 is ACG, codon 3 is GAG, and so on and so forth. Now that codon is critical because that's how we're going to read the DNA in terms of the protein sequence. And in order to do that, you have to use a codon chart. So codon chart or codon tables are used to decode codons. So those codons correspond to what are known as amino acids. And here are 2 tables. Here's one table. Here's another table. They're both used to decode the mRNA message. And they look different, so you may see either one on test A, but they both are used in a similar way. And we're going to run through that now. So the genetic code is important again because it's universal in all organisms. So again, back to our benchmark here, the genetic code is critical because it's universal in all organisms, meaning that these base pairs are highly conserved among species. So that gives us evidence towards organisms possibly having a common ancestor. Again, there's a benchmark there that you definitely want to review. So here's our example. Starting with one side of a DNA, right? You have your DNA template. We have the sequence TAC, TAG, CAT. Now that DNA sequence would be transcribed into an mRNA copy. Again, that would transcribe into AUG, AUC, GUA. Notice A and U pair together in mRNA. Okay, so this is protein synthesis. Now, how do we read this mRNA into a protein code? Again, we have to use our codon chart. So starting with the first three base pairs, AUG. Looking at chart 1 here, you start with the inner circle would be your first base pair. Your light colored yellow would be your second base pair, and your third, your dark yellow would be the third base pair. So if our first base pair is A, we're going to start with A. Our second base pair is U, and then our third base pair is G. So we see that the first amino acid is methionine, and that's what we see right here. Same thing in the second codon chart. A is on the left, U is down the first row, the first column, and then G would be right here. So again, methionine. So practice locating codons on that chart. Our second codon would be AUC, and that codes for isoleucine. And you see here, A is our first letter, U is our second, and C right here is ILE. And then GUA codes for valine, that amino acid. First letter is G, U is right here, and then A. There you go. So in this sequence of DNA, we have 3 codons, and they code for 3 amino acids, or a protein in this case. So in terms of mutations, now sometimes a cell is copying thousands upon hundreds of thousands of base pairs. Sometimes those base pairs result in mutations, and they're just mistakes that happen during DNA replication. So those mistakes can happen during replication because the cell is copying so many base pairs. It could be skipped, or a mistake can happen. And other times it could be due to something called a mutagen. And a mutagen is anything that causes a change in the DNA. Something like pesticides or pollution or radiation are all examples of mutagens. Now back to our benchmark, we're trying to see exactly how do these mutations result in changes, in phenotypic changes, and that's related to its protein sequence, just like we've been illustrating here. So you'll see in this example, we've got an individual that's got a normal DNA, and you can see here is his mRNA code as well as the protein code. So using the codon chart we just illustrated in the last slide. This individual has a mutation, and you'll see here it occurs right here. This base pair in that mRNA, which originally came from the DNA, was replaced G with an A. And that's important because if you look at your codon chart, the sequence AGC no longer codes for glycine like it should have. It codes for an amino acid called serine. And serine, now being in there, will change the sequence and thus cause the mutation in the protein. So that's an example of a negative change. But I'll give you an example now of a change that may not result in a mutation. If you take a look here at that, again, normal individual, and at this sequence, GGC, if this base pair here, C, was converted to an A, then you'll notice by looking back at your codon chart that GGA codes for glycine also. So that individual will still produce the correct amino acid in that sequence, therefore not resulting in any change. So I hope this helped. Lots of luck on your exam, and best wishes.